Okay, question number 9. There are lots of good techniques in here, and we just need to be clear on these conversions of units, which was really unfortunate. We do look at this when we do microscopy. Okay, so the smallest DNA molecules are very long. We've got a kilobase as a unit equivalent to 1,000 base pairs. 1 kilobase has a length of 0.34 micrometers. Okay, so this little sign means that it is a micrometer. Okay, and we've been given 5.6 centimeters. So in 1 centimeter, you have 10 millimeters, and then in 1 millimeter, you have 1,000 micrometers. Okay, that is on the syllabus. There's a table where we need to learn these conversions. That's the main one. Then we do actually go down to nanometers, which is another thousandth of a micrometer. So in order to do this conversion, we've got 5.6 centimeters. We need to convert that into millimeters. That would be a multiplication by 10, and then into micrometers. So if we multiply this by 10,000, then we'll get the micrometer size. Okay, so the DNA nucleus of the cell from reply is 56 micrometers long. We're going to divide that by 0.34 to work out how many kilobases there are in there. And you should have got 164,706 kilobases in total. Okay, and remember that is still, you need to times that by 1,000 again to get the number of base pairs. So huge numbers here. The first thing is, hopefully just try and think about the boundary of this. You know this should be a big number because it's told you that the DNA molecule is very long. If you got this conversion incorrect, don't worry too much because, like I say, we go over it in another topic. But if you had some form of, 5,600 or 5.6 divided by 0.34, and you've shown you're working, you could still pick up a mark there. Finally, the DNA of the fruit fly is analysed. This was done fairly well. So this refers to something called Chargaff's rule, which we did talk about. So 22% of the bases were adenine. Therefore, logically, because of complementary base pairing, we know 22% must be thymine. 22 plus 22 is 44%. So if you've shown that you know 100 minus 44, that leaves us with 56% remaining, which divides that by 2 should be 28% is going to be G and 28% is going to be C. Just a few people, I know, having a bad day and thought that 100 minus 44 was 66. But I could still see the logic, so you could still pick up a mark. Okay, and we actually looked at these questions a little bit in today's lesson. So we've got condensation reaction releasing water, and then a DNA molecule contains 2 polynucleotide chains. How are they held together? Just have a sort of visualize it there. You've got these 2 polynucleotide chains with our bases going across the middle. So those are going to be hydrogen bonds. But actually, this is your clue, polynucleotide. So also, how are the nucleotides themselves held together? So you could get a mark there for phosphodiester, which not many people wrote, which links up that sugar phosphate backbone. You could also get marks for saying there's H bonds. And then you get 2 extra marks for elaborating on that. So hydrogen bonds, which hold the purine to pyrimidine, or you could say A to T and G to C. And then also for mentioning the bond numbers. So saying that there are 2 holding A to T together and 3 holding G to C. So I think that's a good example of a question where you can still get it right if you sort of slightly misread the question, but realising the importance of that word polynucleotide will help you just reach those extra marks.